Hello everyone, my name is Jose Hernandez-Monterroza. I am a master's student at the Horticultural Science Department in UF, and today I'm gonna talk about understanding physiological roles of initial leaves of bare-root strawberry transplants during establishment and their impact on field performance. My advisor is Dr. Shinsuke Agahara. To begin with, strawberry production has been lead for these 10 countries. In 2017, they were the higher producers, being China and the United States the biggest one among all them. And the strawberry production in the United States in 2018 showed that California was the leading producer with 90% of the total production, and Florida being the second one with 9%. In 2017, the farm gate value of strawberries in Florida was 337 million, being the number one among all berries, vegetables, and melon crops. As you can see in this graph, Florida production, price, and early planting. The red bars mean Florida production. The blue line means the price per flat. What is important here is that growers in early December, they can get higher prices for their berries, about $17.84. But later in the season, the price dropped down as Mexico starts exporting a lot of strawberries to United States. And something that we have to say is the production is minimum or little in the beginning of the season, but it increased as the season goes and continues. And therefore, planting has been pushed back, very early planting, for example, in late or middle of September. This is the strawberry production timeline in Florida. Establishment goes from October to November, and very important, the early harvest is middle November to December. Bare root transplant production to farm. The bare root transplants, they are produced in north states, for example, California, North Carolina, and also Canada. The bare root transplants are harvested during September and November from 4 p.m. and 4 a.m. in the nurseries. Then they are sent to the sorting houses where they cut the excess of root and stems. And for some states, they cut the leaves as well, but not for Florida. This is different, and we're gonna talk about it soon. And these are the cooling facilities where they store them at 1 to 2 degrees Celsius. And they are shipped within 20 hours after harvest, and they are planted in the field as soon as they get or as soon as possible. Transplanting in Florida goes from September 20 to October 15, and this is how we get our transplants in Florida. Growers have long believed, and by experience, that having initial leaves in the bare roots promote quicker establishment in the field. So that's what we are testing in these experiments. We receive them in boxes of 1,500 plants per box, and after transplant them, we do this in the morning, very early in the morning, so we minimize the stress for the plants and also for the people outside working. And sprinkler irrigation is immediately turned on after transplanting to keep them hydrated, and it goes from 8 a.m. to 6 p.m. For 10 to 14 days. These are the desirable transplant characteristics. We need undamaged leaves and stems. Very, very important what we are gonna see in this presentation. And large crown diameter, minimal dehydration, and sufficient and functional roots as well. Why are initial leaves important? So for this experiment, we developed 3 treatments. The control, we didn't do anything to the leaves. They remain untrimmed. And the untrimmed but cover treatment, which we cover them with aluminum foil, the leaves, in order to prevent the sun to they can produce photosynthesis. And then the trim treatment, we cut the leaves about 2 to 3 inches from the base of the crown. And our hypothesis here is that initial leaves promote the new growth by translocation. It means moving nutrients from the initial leaves to the crown, or doing photosynthesis, producing new carbohydrates to support the new growth, or by doing both, translocation or photosynthesis. Some concepts that we're gonna see in this presentation, for example, new leaves, we say that those are the ones that develop after transplanting. The mature leaves are the ones coming after the new leaves. They are fully developed and they become mature leaves. And initial leaves are the leaves that the transplants come with from the nursery. Similar to initial roots, those are the ones that the transplant have, and new roots are the ones developed from the initial roots, from the tip, or from the crown. So the objective of this study was to understand the physiological roles of initial leaves of bare root strawberry transplants during the establishment period and their impact on field performance. For this, we conducted 2 experiments, one greenhouse experiment using a scanner-based Rhizotron system, and a field experiment to examine plant growth and yield performance. Materials and methods. The experiments were conducted here at GCREC in Balm, Florida, and the experiment period was conducted from October to November 2018. It was conducted for 34 days. The experimental design was an RCBD with 10 replications. And for the field experiment, it was conducted on October 2019 to March 2020. And the experimental design was an RCD with 4 replications and 18 plants per plot. The material that we used for this experiment was the cultivar Florida Radiance. This is how the Rhizotron and field experiments look. This is our Rhizotron container. This is how we scan the root data. And this is our field experiment. For the Rhizotron experiment, we collected the soil from the field that is already fumigated and fertilized. And also we pack the soil using the soil bulk density in order to have same conditions for the soil in the Rhizotron similar to the field. And Rhizotrons are inclined at 30 degrees to promote the root growth on the bottom side of the container. And this is the picture that you can see. And the Rhizotrons are covered to keep the roots in the darkness with these 2 shade covers. For the data acquisition, this is how we get for the canopy area with a digital camera. This is how the photo looks for the canopy area. And with ImageJ program, we calculate the canopy projected area. Similar for the root data, we scan the Rhizotrons, both sides of the Rhizotrons with a top bench scanner using a tablet. And when we get these photos, we analyze these pictures by ImageJ as well to get the root projected area, or we can use wind Rhizotron to estimate other parameters like root diameter, root length, and so on. The Rhizotron experiment results. So as we can see, the canopy growth in these images for the untrimmed cover and trim treatment from 6 to 20 days after planting. As you can see in the untrimmed treatment, canopy is larger compared to the canopy in the trim treatment and the untrimmed cover. And in these videos, you will see the root development in this treatment as well. The blue line is the roots developing from the crown, and the green lines are the root developing from the tip of the initial roots, as you can see in this video. And it goes from 6 days after planting to 34 days after planting. And this is the video for the untrimmed cover and for the trim treatment. as you can see in the images or in the video, it's very clear that the untrimmed treatment developed faster and larger root development compared to the untrimmed cover and trim treatment. This is very important because it means the initial leaves are doing something in here. And this is what we get in the data. As you can see, untrimmed treatment perform better than untrimmed cover and trim treatment. And as we can see in the canopy projected area data, that untrimmed treatment in the blue line and orange for UTC and black for the trim treatment. As we can see here, at 6 days after planting, UTC had 68% and the trim treatment had 87% less canopy projected area compared to untrimmed treatment. And at the end of the experiment, it was 24 and 54% less canopy projected area compared to the untrimmed treatment, which is telling us that initial leaves have a big impact on the canopy development in these treatments. And we had a similar trend for the root projected area. As we can see, UT or untrimmed treatment performed better, having a larger root projected area compared to the other two treatments. And as we can see at the end of the experiment as well, it had for UTC 45 and for trim, it had 66% less root projected area. And for the photosynthesis and stomatal conductance, we can see in the initial leaf for the untrimmed treatment that this is very important because we can see that initial leaves are producing photosynthesis even from the beginning and improving its photosynthetic rate until it reached to 13.4 micromoles, meaning that they are producing new carbohydrates to support the new growth in the plant. And this is what we are seeing in this data as well. For the field experiment results, this is the initial leaves. This is the setup for this experiment. Initial leaves are very important, as we saw in the previous data, and we are confirmed in the field as well. Untrimmed performed better compared to the trim treatment, but there was no difference between untrimmed and untrimmed cover treatment. Initial canopy growth and early yield, as we can see in the untrimmed and trim, untrimmed treatment performed better with canopy projected area compared to the trim treatment. And what is more important here is the early yield. As we can see, untrimmed treatment in the blue and trim in the black line, we have that about December 30th, the trim treatment had 81% less early or yield compared to the untrimmed treatment. This is a huge difference and it's very, very important for growers, as we can see in the data. And this is photosynthesis and stomatal conductance in the field. And this is similar trend for the initial leaf. And something important that I just want to mention quick is that new leaf and mature leaves, they exchange roles during the seasons. At the beginning of the season, new leaves produce more photosynthesis where there are no mature leaves. But as the season goes on, new leaves decline in photosynthesis because they change the role. Mature leaves produce more photosynthesis than the other ones. As conclusions, initial leaves enhance canopy growth and root development during establishment by supplying photosynthesis for new growth via both translocation and photosynthesis, but translocation appears to play a more important role. Improved establishment growth supported by initial leaves can increase early yield, which is critical to the profitability of strawberry growers in Florida. And as a recommendation, minimizing transplant damage and dehydration between a nursery and production field is key to the production, early yield production. And I just want to thank my lab for all their help and support, especially my advisor, Dr. Shinsuke Agahara, my committee members, and all my team. I am available for questions. You can contact me. These are my email and my personal email. And you can also scan this QR code, which will send you to a quick or prompt to email me quickly with your questions. Thank you very much for your attention.